Hello everyone. In this video, we are going to discuss about limit test. So what is limit test? Limit tests are nothing but this is a analytical technique used for identification of impurities present in the pharmaceutical or chemical substances. This is the general in the general sense we have to say that identification of impurities, that is called limit test. Okay. So, in broad science, we have to say that limit test is defined as the quantitative or semi quantitative test designed to identify and control small quantities of impurity which is likely to be present in the substance. So, this is the broad definition. Next, come to importance of limit test. So, why it is important or importance of limit test? To find out the harmful amount of impurities. Yes, this is the very important thing. If any impurity is present in the chemical substance or the pharmaceutical substance, that will interfere in the assay of that particular substance or purity of that substance. So you have to perform limit test to identify the impurities. Next come to find out the avoidable or unavoidable amount of impurities. Then most of the limit tests for chemical substance or pharmaceutical substances are performed using Nisler cylinders. Yes, in case of limit test, the glass apparatus those we are using that are termed as Nessler's cylinder, and these are made up of borosilicate glass. Okay, maximum laboratory glasswares are made up of borosilicate. Okay, they must be calibrated. Always be remembered that one. Next come to general procedure for limit test. Any limit test is there is a simple procedure for limit test is involved in any type of limit test, whatever you have to perform. Okay. Next come to procedure. First, there are two Nessler cylinders should be taken. One is regarded as test and another one is regarded as standard. And here I have draw the two pictures. One is test and standard, both the cylinders, and I have written that 50 ml on both the cylinder. That means the available Nessler cylinder in the laboratory, they are labeled or they are labeled as 50 ml. The capacity of that cylinder is 50 ml and 100 ml also available. So you have to make the volume in that particular Nessler cylinder of this test solution and standard solution 50 ml. Okay. Next come to test cylinder comprises of sample to be checked for impurity that is unknown. So the sample to be tested that is unknown. The standard cylinder contains known quantity of sample to be tested as standard. Okay. So that's why that designated as standard and the left side cylinder is designated as test. And limit test involve the simple comparison study of opalescence, turbidity or color produced in test with that of the fixed standards. Okay, so this is the broad or basic principle for limit test. Okay, thank you.